CellSorter Scientific has a technology for automated cell picking and single-cell manipulations on a microscope. We develop, engineer, and supply technology for image-controlled single-cell manipulations. You can target cells directly based on their morphology and fluorescent signal. General features of the instruments. It can collect single cells for further cultivation, cloning, DNA, RNA, or protein preparation. You can sort cells directly from the Petri dish or plate. The device is compatible with 24, 96, 384, and 1536 well formats. Cells are viable after sorting. The instrument is modular and upgradable. The average sorting process takes only a few minutes, less than half an hour. Cell culture needs minimal preparation before sorting. Both unlabeled and fluorescent cells are recognized by computer vision. It can isolate a subpopulation of cells expressing fluorescent or luminescent markers. Adherent and suspended cell types can be sorted as well. Multichannel detection using the fluorescent filter cubes of the microscope. We have 3 products. The QuickPick system was launched in 2013. It is a versatile instrument for research labs with several years of experience in many applications. The NanoPick head is our second-generation technology. Nanoliter precision is ensured by a patented piezoelectric micropipetting technology. We constructed a compact desktop device, NanoPick 1000, optimized for high-efficiency single-cell picking. Our entry-level instrument is the highly flexible QuickPick system. It is a semi-automated setup compatible with most inverse microscopes including Zeiss, Nikon, Olympus, and Leica. The micropipette is held by a rotatable arm attached to a high-speed motorized micromanipulator. The robotic arm can be easily removed and reinstalled if the microscope is used in other applications. The LED illumination of the micropipette supports the perfect alignment of the tip. The NanoPick add-on head is a unique technology with a granted patent family. The add-on version provides fully automated single-cell picking on a standard microscope. It comes with built-in LED illumination for pH 1 and pH 1 phase contrast imaging. You do not need tubes or syringes anymore. Liquid handling is directly controlled by electricity. NanoPick offers sub-nanoliter liquid handling accuracy. NanoPick 1000 integrates the NanoPick head in an encased cleaner environment. It is an optimized, fully automated small footprint device for single-cell picking with 6 fluorescent channels. You can also use the joystick to manually control the stage or the micropipette. We offer a range of sample holders for Petri dishes, PCR tubes, and standard plates. Flow cytometer is the well-established method for cell sorting. We compare our technology with flow cytometers. Our technology provides a compromise-free imaging power instead of the simple fluorescent intensity of cells or the limited imaging capabilities of high-end imaging flow cytometers. We believe that the high-resolution image of cells contains far more information than just the fluorescent signal. Sensitive cells can be damaged by the sheer stress of flow cytometers. Picking cells gently in a few nanoliters minimizes cell stress and enables the handling of precious cells. Sub-nanoliter precision ensures efficient single-cell picking from suspension. High-throughput imaging allows the scanning of a large number of cells in multiple channels. Rare target cells recognized by artificial intelligence can be isolated from millions of other cells. The operation cost is low with a single disposable component, the glass micropipette. The device's cost is very competitive. We have users worldwide. Most of them are in the United States and Europe. We also have some customers in China, Japan, and Australia. Our users are top university and research institutions including MIT, Boston, Karolinska, Stockholm, Institut Pasteur, Paris, ETH, Zurich, and more. The workflow of the standard protocol is straightforward. The cell culture in a Petri dish or multi-well plate is scanned on a microscope to gain a large mosaic image. The images are analyzed by the software to recognize the target cells. Cell selection can be reviewed and manually modified. Selected cells are picked and deposited by the micropipette into PCR tubes for downstream analysis, for example, single-cell transcriptomics. Our solution can be applied to isolate cells for single-cell transcriptomics. Virtually any cell type can be used, including red blood cells, muscle cells, and neurons. The aperture of the micropipette can be chosen in the 5 to 120 micrometer range to fit the cell size. In custom applications, even smaller or larger micropipettes are supported. After the automated cell selection and isolation, you can run PCR and RNA sequencing. DropSec is a droplet-based single-cell isolation and DNA or RNA library preparation method for sequencing thousands of cells. Our approach is different. We start with imaging the cells to understand their phenotypes, including protein-level information with antibody labeling. We have a detailed morphological information of cells, including protein localization with appropriate fluorescent markers. Cell behavior can be followed in the time-lapse imaging mode. We spare the high cost of library prep and sequencing of many cells by selecting the target cells before these steps. Protein engineering can be assisted by single-cell picking after random mutagenesis. The best performing protein expressed in a cell is selected for isolation. The cell is lysed for whole genome amplification, PCR, and cloning to identify the target gene. The method can be combined by flow cytometry as a cell enrichment step to exclude under-threshold cells before cell picking. Rare cells can be detected by image analysis and isolated by the robotic micropipette. Applications include circulating tumor cell analysis from a blood sample, fetal cell investigation from maternal blood, and rare brain cell research. Genomics and transcriptomics of the isolated rare cells can be characterized with PCR or next-generation sequencing. Cellular adhesion analysis. You can probe single cells' interaction with surfaces decorated with biomolecules. Run a functional measurement on live cells. A straightforward and high-throughput technique. Hundreds of cells in 30 minutes. Investigate drug effects. It is a high-throughput alternative to AFM measurements. The technology is capable of the functional analysis of intact immune cells. You can investigate cell surface receptors or probe the interaction of endothelial cells and neutrophils. You can also generate hybridoma cell lines by picking the best performing clone. We offer online and on-site demos worldwide. We have 10 minutes video tutorials to train new users. We provide installation and hands-on training. We can assist you to optimize your specific assay. We are always happy to help when you need scientific support from our inventors and developers. We can customize both our software and hardware for new applications. Our publications appeared in the field of single-cell picking, manipulations, and isolation. Our users published scientific papers introducing new applications. Protein engineering can use our tools for creating new proteins with enhanced features. Cellular adhesion force characterization is a field of biophysics with relevance in tumor biology, embryology, and tissue engineering. Cellular immunology can be studied by probing the adhesion force between immune cells and other cells or a solid surface decorated by antibodies or other immune molecules. Single-cell transcriptomics emerged when next-generation sequencing techniques enabled the analysis of RNA from individual cells. Rare cell isolation and molecular characterization can be useful for liquid biopsy to detect and investigate circulating tumor cells. Another medical application can be non-invasive prenatal testing, NIPT, for screening the potential anomalies of a human fetus from a simple blood test. Thank you for your attention.